Welcome to the Microscopists, a bite-sized bio podcast, hosted by Peter O'Toole, sponsored by Zeiss Microscopy.
Today on the Microscopists, I'm joined by Christophe Leterrier, team leader at NeuroCyto in Marseille and director of the Nikon Center of Excellence for Super Resolution Microscopy. In this episode, Christophe reflects on the make or break moment at the end of his postdoc, when he came close to leaving academia for good before securing his role in the CNRS, and how that experience now shapes the way he mentors others. He also shares his thoughts on preprints and how they've transformed the pace and openness of publishing, and what challenges remain as the system continues to evolve today. All this and much more on The Microscopist with Christophe Leterrier. Hi, I'm Peter O'Toole from the University of York, and today on The Microscopist, I'm joined by Christophe Leterrier. Christophe, how are you today?
Fine, thank you. Thank you for having me.
Oh no, it's more than a pleasure, actually. I've not met you very many times at all, so I'm really looking forward to get to know you better through it all. So I'm going to, where do we start with this? Maybe I'll start by, actually, what is your current role? There you go, let's start there.
My current role. I am in Marseille, France, and I lead a lab there that's called NeuroCyto for the neuronal cytoskeleton. And it's a lab that I've started maybe 2017, so soon it will be 10 years. And so it's in an institute that's called the Institute for Neurophysiopathology, which is fundamentally a neuroscience institute, although what I'm doing might be the more cellular side of neuroscience, or even say it's more cell biology than neuroscience. So I have this role as a team leader and driving my research and training people. And I also a role as a facility director, because I have set up an imaging facility at the institute that is specialized in super resolution microscopy for neuroscience. And we are lucky to be a Nikon Center of Excellence. So I am taking care of this and trying to bring these techniques we use to as many people as possible.
Do you know what? I am missing right this moment Nikon training on our new spinning disc. Just come in as we speak. We're getting trained on it. I say my team are getting trained. When did you start? When did you set up the facility?
So when I was back in, so before we moved to our current location, which is Marseille Center City, I was near the hospital in the northern part of the city between Marseille and Aix. And there was a very small facility with like 3 microscopes. And at some point I started taking care of them, and I think I was taking care of it for about 5 years, but that was really a small operation. And when we got the move, I was lucky to be involved in thinking about how we could develop that in a more comprehensive facility. And so the facility really started with the move, so that would be 2019, and we got a big equipment operation that allowed us to buy 4 different advanced microscopes in 2020. So I would say 2019, 2020 is really where it started for the facility.
Did you become a Nikon Center of Excellence at that point, or was that later on?
It was the big equipment upgrade that included some Nikon microscopes that set up the Nikon Center of Excellence. So of course that was COVID, so all the timelines are a bit blurred. Formally the Nikon Center was inaugurated in 2021, but yeah, that was around that time, and from this push. To get and to equip the facility with several types of super resolution microscopes.
So does that make us rivals if we're a Zeiss Labs at location site?
I'm sorry?
So does that make us a rival, being as I'm a Zeiss Labs at location site?
No, not really. I mean, I think the landscape of super resolution is really broad now, and there are really good technologies, mature technologies from different manufacturers.
How do you, this is interesting. So obviously, you've got your academic group with it. How do you separate your academic side to your running a core facility side? Because one's very academic, there's teaching, lecturing, there's the academic research, applying for grants, but then there's a core facility, which is a service. Arguably, it's almost it is a business side. They're two quite distinct things. How do you marry those together or separate them even?
Yeah. So first thing, I don't teach because I'm CNRS, so a bit similar to NIH employee, I would say.
Mhm.
So it's pure research position. So that is a chance. I mean, at least it gives me more time to do research and to take care of the facility side of things. And I would say that for me, the facility started a bit as an extension of what were doing in the lab. So I would say very briefly, it's about using the most advanced available technique and apply them to biological problem and cellular neuroscience problems. So of course the orientation, the general taste of the facility was biased by this. But then after a while, the idea, and this is where I am now, which is really like, okay, I built this tool that is really nice, that is very good for my team and a few teams around, but now I would like it to be much more useful. And I was very lucky that we were joined at the beginning of the year by a very experienced facility manager, Bertrand Vernet from IGBMC in Strasbourg, and that really wanted to take the challenge of bringing that facility to the next level. So this is really what it took a few years to really, know what we were doing, know what we are good at and what we could propose to everyone. But now I think we have a good idea. And so I rely a lot on Bertrand to develop these kind of things. And I am really more in like an academic supervision, like, okay, where is the facility heading? How can we serve the user? What kind of microscope can we upgrade or things like that, and not to the day-to-day operations.
Yeah, so it's good that you got someone like Bertrand actually in there as a wing person to actually look after it and
Yeah.
do a lot of the admin, the bureaucracy, the just looking after the users. Which side do you prefer, the academic or the core facility?
Uh, I think what I used to prefer was to just spend days in front of a microscope, with nothing else to take care of. But these days are quite over for me in any side. So I would say, uh, I don't know. I think right now it's more the academic side, but as I was saying, I'm getting more involved in the facility right now because I really want to push it and to make it available and useful for a lot of people. So I would say in the recent year it was really establishing my group because
Hmm.
that's a hard thing to do and really pushing the projects and publishing the papers and getting the grants. And now that I have reached something that I would see as more like I would say yeah, a level that I would be happy to stabilize, let's say. Then I'm like, okay, now maybe the next challenge is to spend a bit more time on the facility and try to help people with what we know.
I'm just thinking back to conversations I've had with different people and thinking, oh, this is getting established, it's going to plateau, become stable. It's never got stable, I've
It's
got to say. And microscopy is just the growth within microscopy is so big. It just grows and grows and grows. Do you really think it's going to get to that point for your research or the facility?
no,
Because
no.
I think honestly, they're just going to keep going on upwards trajectories.
That's exactly true, and I think that's the main challenge because I mean, this is exactly the point where I'm at right now, looking at the future and saying, okay, there is this natural slope. I would say like, okay, everyone expects you to always grow. Everyone expects your facility to get bigger, to hire more people, and the same in the lab. And it is extremely difficult in the way things are the system work to say, okay, I have a team of 10 people and I'm perfectly fine with it. And now I would like to just, try to bring my ideas to life. And with this team, that is a size that I can manage and that I'm comfortable with because they will be like, oh no, this little grant, you already had it. Now you have to aim for the bigger grant, or you have to try the bigger microscopes and I think personally, I think like I have to kind of devote efforts to not do that. Like to say,
Mhm.
okay, no, I really would like to not play that game of trying to get always bigger. But I agree that this is what everyone expects. So I don't know if I will manage. And I mean, if the worst thing that can happen, if I get bigger and I have to play that game, then I will not be unhappy. But I think that at some point it's important to know what is the right size and the right span you want to have.
Yeah, no, I actually just put that on mute. I've had this conversation recently with people internally, the importance of choosing the right equipment and not what everyone wants. Because what one person wants may just be for one person. Even
Yes.
though lots of other people might say they would use it, you've got to really look at their science and think, well, A, do they really need to use it? Is it going to benefit them? Because we have to sustain these. And it's, as you say, there's a lot of chocolate bars on those shelves to buy. And you can't eat them all. Because if you do, there's not enough people for a bar eat. It dilutes out quite quickly. Do you have problems with other academics? Have you ever been accused because you've got your academic research of choosing what you need for yours and ignoring what others want? There's a difficult question.
In a way, I think I was a bit lucky to be in an institute where there were no other very strong imaging inclined people or teams. So basically, there would be like, oh, you have these nice things, that's perfect. Have fun with it, and as long as we have our good confocals, we are fine with it. And this is exactly why now I'm like, okay, I was lucky to be able to design a bit the facility around the kind of research we do, but I am convinced that this can be much more useful than that. And this is what I'm trying to do now. And to look back to your point, it's always difficult because it's always a constant feedback between the needs of the user and what you can propose and the fact that you have a new technology or something that can bring something to the table might generate a new need from the user. And you have somehow, as you say, to look at the whole situation and say, okay, because as I'm sure you know, if you ask what people want, they will buy 20 spinning disc microscopes. Because what they want is to have one just for them that is always
Yeah.
when they need it. And there is like the current thing that everyone likes, and they will just want to have that forever and in 20 different setups. So you have somehow to bring new stuff and to show people, and this is where having good people in the facility that are really able to convey the advantages of some technologies and in the other direction too, like to kind of dampen the marketing, or people that come and say, oh, I need to do lattice light sheet imaging, and so like, okay, sit down, what do you need really, what
Yeah.
is exactly your sample, what is the resolution you want. And so really and it's this constant conversation between what is available technologically and what the people want to do, and they are both driving each other. So the important thing is to have the conversation.
How did you, yeah, how did you get into microscopy? How did you fall into your academic career? So maybe no, do you know what? I'm going to take you way back to when you were a child, 7, 8, 9, 10.
Yeah.
You may have heard this question before. What was the first job that you ever wanted to do? What was your childhood? I want to be that job.
I I don't really know. I mean, I think I'm not the kind, I heard so many of these interviews where people like, when I was 5, I would try to disassemble the radio and understand how it's the, or
Richard Henderson.
I would try to mess with ants and see where they would go and things like that. I don't remember. I was mostly happy reading books, on my own when I was a child.
Mhm.
So I was reading yeah, knowing stuff was really my passion, and I didn't really know what kind of occupation or job would allow me to do that. I mean, later I discovered I was being an academic, but I would say it wasn't like something I knew for a very long time. And as I was lucky to be quite good at school, my parents who are not academics or don't really know this world didn't know exactly, but they were always telling me, try to do the best you can. So if there's one thing they were really pushing me is to really try to do my best. And they were convinced I was really, really bright. So they would always say, I would come with very good grades, like, I'm sure you can do better. So they were pushing me like that. And this is how I ended up, doing these so in France, we have this strange system with preparatory classes and elite engineering schools and things like that. And these are supposed to give you the best opportunities if you are good at school and good at math and things like that. So I went into that not really because I was like, oh, I want to do this. I want to be an engineer. I want to be to build bridges but really more like, okay, this my parents tell me this is the best I can do. So let's try that and it took me a bit more time and to be a bit more mature while I was in those classes and later in engineering school. I was lucky to be in a school that had very strong ties to academic research. There were like a lot of labs on campus, and this is where I discovered, I would say, what was really for me. There was like hanging out in labs and looking at everyone that did just care about knowing stuff and trying to find something nobody has ever found. And this is so it wasn't like a child, I would say dream, but at some point I would say, okay, I don't want really to work in a company, and make, even if I still have, we can discuss about that. I still have that engineer point of view. I really like machines and even when I do biology, I try to understand cells like little machines. And if you add that and remove that and see what happens. But this is where, yeah, I was drawn to academia when I was in engineering school.
So what was your first degree?
Sorry?
What was your degree in?
So my degree, engineering in chemistry and physics, and so at the end, last year of the school was like, okay, most engineering schools in France are 3 years, but this one was 4 years because the last year you could basically do whatever you wanted. So they say, okay, you want to do something after. There is 1 year where you can do a master's, or the people that really like business could do an MBA, or really the idea was to complement your training in engineering by something that would help you for what you want to do next. And this is where I started to do biology seriously, and I did a cell biology master's during the last year, which allowed me, as soon as I graduated from the engineering school, to start a PhD in neuroscience. So that was my pivot to academia. Was at the end of engineering school, I went into a PhD in neuroscience.
Yeah, where was your PhD? Where did you study?
So my PhD was on the engineering school campus. So the
Okay.
school is called ESPCI, which means physics and industrial physics and chemistry
Hmm.
school. But there was a lot of biology that was developed at the time there, and there were two biology labs on campus. And one of them, I did a 3 month internship as part of the curriculum, and I really liked it. I had the luck to do the internship with a young researcher that was just starting his group and his research, where was called Jolt Lenke. And so after that, I did my my master's internship with him, and I did my PhD with him. And I was his first PhD student, so things were really like, we were starting,
Good.
and it was a great time. So I did my PhD in neuroscience there at the ESPCI lab in Paris.
So I've got to ask, biology or chemistry?
Biology.
Chemistry or physics?
Yeah, I mean,
Which
uh,
one?
chemistry or physics. I would say now more physics. Yeah,
And physics or biology?
biology.
Ah, well done. But I think your chemistry and your physics probably help you quite significantly in the bioimaging world.
Yeah.
Lots of biologists, biochemists, biophysicists, who, as you said, I think you rightly say, I think similar to me, you see it as a machine, you see it as visually of lots of mechanisms going on and physical mechanisms almost as tools or instruments within
Oh,
it.
I think microscopy is really the perfect intersection of biology, physics, and chemistry, because you have the biology, of course, you have the physics, the optics, and all about the also the processing and everything, that is pure signal. And you have the chemistry because you have the sample and you have the sample prep. You have to label your sample, and so you have to also understand a lot about the chemistry. So for me, it's really the ideal place to benefit from all the training I got in those three disciplines.
So change of tack slightly. Okay, so as a child, no big job. I say I wanted to be a footballer for a fair few years. Obviously, completely useless at it, but you want to be a footballer, I guess, or I did. Uh, if you could do any job in the world for a day or a week,
Yeah.
what would you like to sample? What would you like to try out?
That's a good question. I think if I wasn't a scientist, and if I wasn't doing what I'm doing, I would like to be an architect, because I
Oh,
think for me I mean, because this is also the way I see a cell, as a building, as something, that is made from pieces, and that is also at the intersection of something that is very, very scientific and something that is a bit more artistic.
I'm just trying to think through thinking about the mechanisms, but we think about it as tools, but it's all very fluidic and very motile. Just wondering now how you could architect something that is so fluid, but I'll let you think about that. You want
Oh,
to
but
be
I
the
mean the
architect.
the the, cons- the building process. Of course, once it's built, it's not supposed to move, but the way you assemble it so that
Ah,
you end up with the structure.
but wouldn't it be cool if it could? Ah, just a thought. If I now come through to what has probably been the most difficult time in your career?
Uh, the most difficult time. Hmm. I think the most stressful was the end of my postdoc before I got a position as a researcher, a CNRS position, because I really felt like I was this close to drop out. I applied, and I had an audition I had several auditions, and nothing worked. And so I had one more year where I could try, but I somehow knew it would be my last chance. And if it didn't work, then there would be no stable perspective for me in academic science in France. So I would really have to choose, like, okay, I want to go somewhere else and try to be an academic outside of France, or do I want to just stay here and do something else? And that was really like the crossroads, and that was quite stressful. And it ended up working out at the last minute and the last chance. And this is a constant. I mean, this is also in my career. It's like very often I'm not an early starter or someone who was there very early or that got stuff. So I got a CNRS position a bit late, and then 5 years after that, I was really my last chance to get a grant to start a group. So I started a group, but I was already almost 10 years after my PhD, and and every time I would feel like I was sneaking, like Indiana Jones under the door, in the movie. And I try not to forget that, because of course once you have made it through, everyone is like, oh, we were sure you would do it. Of course you deserve it and everything, like it was natural. But I never forget that several times I was this close to, for the door to really close, and to not be able to go forward. So now that I am, mentoring people, and on the other side, on the committees that have to decide, who will get a position and things like that, I try to never forget, that it's not so clear what makes the difference between someone you say, okay, this person is in, this person is out, because that could have been me.
Do you know what? There's a good question. When you're looking at the next person who's going to get the next position, do you look at their academic record solely, or do you look at the person themselves and to see if they? I think quite a lot of good academics have got something about them, you can tell, as they say. So it's not just the academic record, because people can get unlucky, but there's a spark, there's a difference sometimes that isn't always there. Some postdocs have got excellent academic records, but are probably never going to fly as their own academic lead. That's not their strength. And others
maybe struggle a bit during their postdoc, but really have that academic vision. So how do you weigh that up? How do you even assess that? Is it just a gut feeling?
Yeah, it's a difficult thing to do. I would say as a team leader, when I hire people, like if I hire a postdoc, this personal view has definitely a role because I mean I try to have long conversations and really I think if I feel really like okay that person I want to discuss with that person every week for hours and we are connected scientifically, then it doesn't really matter if she, you know, of course there is a minimum level of
Yeah.
track record, but then after that, you know, it's more than that. And as you say, people can really just completely transform themselves between the beginning and the end of a PhD or the beginning and the end of a postdoc. So I think it's always worth. Now, if we're talking about like I am on the committee, for example, for CNRS that gives the permanent position. So basically, we are hiring people for life as public servants, and there is a big responsibility. And of course, it's not just the track record. This is why we have auditions and we spend some time discussing with every candidate. And you have 16 of us, 16 researchers around the table. And so that it's not just pure bias from one or two people. So there we try to be objective, but I agree with you. There's always a factor that is someone who leaves the room and everyone is like, wow, that was so great. I wish I would work with that person. You know that this person will make a good scientist, and that's not completely the whole thing because of course the track record and the CV is important, but that's a big part of it because unfortunately, given the small amount of positions we can award, you have to have the CV and you have to convince the people once you're in the room. But I agree that this is important. But as you said, it's not perfect science and we have to make guesses and I'm sure we have missed some great people and that's always but yeah, we really try to make the best.
No, and a very stressful one because, as you say, it could be someone's sliding door moment. It could be their last chance. You
Exactly.
know, where you were and you got through, it could be their last chance. And then, you know, it's quite difficult as it's going through PhD, postdoc into academia. It's a very linear pathway to actually go out of that environment. It can be quite a shock. It's quite a cultural change, to different environments, but it is the world. It is a pyramid. As in most industries, you could be an industry that I guess the same, and you come to a point where you've either got to leave
Yeah.
or carry on up it. Oh, life isn't simple sometimes. Talking of which, when life isn't simple, Christophe, what do you do outside of work? What's your hobbies?
So I do a lot of cycling these days. So I got my road bike, my gravel bike, and now that the my daughters are a bit older, I have a bit more time. So on weekends, I go cycling around Marseille, either in the mountains or on the roads, with my wife also, and we kind of hike with the bikes. And I would say the other thing I do quite a lot is cooking. So I like cooking, and so I like to come home and cook a good meal for the people, and nothing fancy. I'm not into it's not being French is not required to have Michelin stars when you cook. Uh, but yeah, no, really like good food that everyone enjoys, a good burger or a good curry, yeah, I like that. And funnily enough, I think it has really replaced the bench work. If I look at the timelines, it's really when I started being away from the bench and not doing experiments so much that I started cooking more at home. So
You what's your secret? Two things. First, I'm just going to say I admire you for cooking curries. Actually, I find that one of the hardest dishes to get just right. Every now and then I'll strike lucky and it's just as I want it. Other times, I find it a lot of hard work, a lot of tasting as I'm going through it to just balance it right.
yeah.
Really challenging. Certainly not one of my signature dishes. But what is your signature dish? What do you most like cooking and what are you most proud of cooking? Maybe two different questions here.
Well, I would say it's a curry because maybe not anymore. Maybe now it would be more a pasta dish or something more Italian. But when I started cooking in Marseille, the main reason why I could not find a good Indian restaurant because I really liked Indian cuisine, although it's a lot of different cuisines. Let's say Indian cuisine. And in Paris, I would have my really my preferred restaurants where I would go every time I craved. And in Marseille, there was none. Like there were really some of them, but they were pretty bad. And I had a good friend who was from London, a British guy, who was cooking delicious Indian food. And so I said, okay, you have to teach me how to do that. And he said, okay, next time you go to London, I give you the books you should buy. And so I ended up buying a few British books because he told me that French books to learn Indian cooking were rubbish. So, and I started doing that. So that's my background, which has nothing to do with my roots or anything, but it's just that I liked it, so I started cooking for that.
I'm just thinking actually, so you go off recipes and certainly with curries, the quality of the ingredients, the specific ingredients can really vary.
Yeah,
Even from batch to batch can vary.
yeah,
And you said how when you went from the bench, I would compare this almost to expansion microscopy,
yeah.
where
Oh, yeah. Well, you have to have the right, and you're looking at the color of the.
yeah, and that's be just the right ingredients, not even from a different supplier. The same ingredient from a different supplier can completely ruin that type of experiment. That's how
I would say,
specific
yeah, I would
it
say
be.
doing a good curry is not difficult. But yeah, doing a great curry. I mean, the final step to something that is really, really great. I don't think I can do it still. I mean, I'm doing it for 15 years and I'm still going to some places and the first test, I'm like, wow, this is another level. So there's definitely something that has to do with some fine tuning and ingredients and things like that.
You are probably your biggest critic because when you cook for yourself, it's always different, isn't it?
Yeah,
You're
yeah.
doing it
But
for
I
the-
am very so I am absolutely not a vibe cooker. I mean, it's exactly like at the bench. I really need to have a recipe, an exact ingredients and quantities, and then if I have done it 3 or 4 times, I consider I know the baseline, so I allow myself to stray a bit apart and to test. Exactly like you would do in an experiment. You change one thing at a time and you see if that is better or not. Because my fear is always like, I don't have the control. How would I know that it's better or not than the original recipe? So yeah.
No, because I would say even the base tomatoes can be different sweetnesses.
Yeah,
I'd
yeah,
say
it's
the
true. It's
cumin can
true.
be
It's
slightly
yeah,
different. So you have to put a little more, little less. Ah.
yeah.
Anyway, so that's what you enjoy cooking. But what is your signature dish? What dish are you like? That's my go-to. If you were entertaining friends, what is the dish you?
Okay. If I have now if you tell me there's 8 people coming tonight, you haven't time to prepare something very incredible, I would do just sausage pasta. So it's the simplest recipe there is, but you have to do it well. It's just, Italian sausage, caramelized, deglassed with white wine, and then you make a sauce with the pasta water, so you make it creamy, and it's like 5 ingredients. There is
Mhm.
parmesan also, and when it's done properly, and when the pasta is the right size, the bits of the sausage are the right size, so some of them enter the tube of the pasta, the chemistry is there, and that's an incredible dish, and it's very easy to make in half an hour.
Uh, it's not even dinner time for me, and I'm already getting hungry. That's really cool. I presume your daughters like all this food.
Yeah, 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 yeah. So definitely they are my also critics for me. So
Yeah, how old are your daughters?
they are 13 and 16. So and I was lucky that I started cooking when they were quite young, so I could train them to like things and to like spicy food and things like that. So I'm very happy that they like most of what I'm doing because I started to train them early to like it. So
I'm not sure I ever trained mine to like the food. I think there was always one of them would not like one of the dishes, and as a child, then they dig their heels in, don't they, for years and years.
yeah, no,
And they
it's.
come back
I
round.
think this was also their personality that they like exploring
Mhm.
and they were quite adventurous for food. So we were lucky for that.
So I've asked about challenging times in your career. What would you say is the most fun time of your career? If you could relive a year or a month in your career, what would you go back to?
I don't know. I think that's a very difficult question. I would say there were incredible times, like hands-on times when I was like the first time, so I'm working on the organization of the axon along axons in neurons and the cytoskeleton along axons and the defining moment in my career has been the discovery by Xiao Weizhuang, not by me of course, but of the periodic scaffold of actin and spectrin along axons. So these axons that everyone thought, it's just a tube, were actually had a very specific organization of actin rings along them, every 190 nanometers, which I think is really the most important discovery made by super resolution microscopy so far. And so shortly after that, I remember the first time I managed to see it by myself on my sample. And like, to my knowledge, nobody had ever replicated it. Like the paper was really just out like 1 month before, and I got the antibody and I was looking at the organization of spectrin in axons, and I see. So this was with STORM, so single molecule localization microscopy, and so we had this nice software where you could see each point, each localization, appearing progressively during the imaging. So you do an image, and it's 10 minutes or 15 minutes of imaging, and you see the points appearing one by one. And I remember looking, it's completely black, and you see the point, and at the beginning you don't know what the pattern, and then very rapidly it starts to be this perfect periodic bands. I will always remember the first time I saw that, like exactly falling exactly like it was in the paper, and I was like, wow, this is not an artifact, this is real. And I mean, yeah, that was a really for me, and but I would say that's maybe the hands-on time, but I would say my most fun times are, collaboration with others and people like Ricardo Enriquez or Stefan Vassilopoulos or Sobojit Roy, people that I've been collaborating with for 10 years or even more now, and all this exchange of ideas and seeing, like, showing, okay, what do you have? What's your data? Okay, here's the data we have on our side. And I think for me, this is what the fun is in science. I'm really not much of a, let's all do it by yourselves. And, I really need to have this kind of sparring partners and people that help me getting interesting in things and seeing other things and confront the views about something. And I think so. I would say, yeah, collaborating with long-term collaborators, people that have become friends, is the most fun times.
But you've also chosen very good partners there that are very collaborative and open. I
Yeah,
would
no,
say.
of course. I mean, it's because I am quite open to collaboration. So I would say the ones that are still there after 10 years are the ones that also like
Mhm.
it. So it's a way to select.
So, who have been your inspirations over your career?
My
Inspirations and biggest motivators. Slightly different questions.
my inspirations. That's I think for I have people that I admire, but the thing is that when you're in France, I feel like you are a bit disconnected from the elite US system. So you are in a PhD in Paris, and you see these papers coming out, and you have these people, and it's incredible, and you kind of idolize people from the papers. But then you get, you develop, and you get to meet people, and you get to be part of a community, and then people are like, oh, that guy, oh, no, no, he's horrible, and things like that. So it's always difficult because you always have this kind of I would say tension between the I guess it's like, you listen to some music and you're like, oh, these people are really geniuses, and then you discover they're not as good as person as you thought they were. So there's always this kind of thing for me that I would say that the inspiration came from the people that trained me, of course, and Jolt for my PhD, and Bénédicte Dargent for my postdoc, were people I learned a lot from, and I picked, I would say some of it, I said, okay, this is how I want things to be. Some of it, I said, this is how I do not want things to be. But that's also a kind of inspiration, I would say. And I would think that there were both in different ways people that were quite exceptional, and I learned a lot, and I have maybe being with these people that were quite different in my PhD and my postdoc also taught me that there are several ways to manage to succeed in science. Like there are people that are very rigorous, and thinking and very rational, and people that are more like you don't really know how the idea comes from, and it's like, and when you hear it, it sounds completely stupid, and then 2 years later, as the project develops, you're like, yeah, that was the right thing right from the beginning, because and you have no idea how that idea came. So I think there are really several ways to, and of course I would say, if I have to say inspiration, and that drove me into what I'm doing, was, these all the papers that basically started super resolution microscopy in 2006. So the people like Eric Betsick and Jiao Zhuang and and Stefan Hell, and all the people that brought super resolution to biology, because that was, I defended my PhD in 2006, and this is when this happened. I was like, yeah, this is completely. I was always and already a big fan of microscopy, but then I was like, this opened, and I was like, okay, and this is why, I started my postdoc. And I wasn't lucky enough to be able to start doing super resolution microscopy right in my postdoc. But then I got when I got my position in 2010, 11, this was, I said, okay, now I have a position for life. I'm in a lab that is well funded. Let's go to do that. And I really spent 5 years getting into Storm and SMLM and trying to do it because of this, the way it had opened my horizons at the end of my PhD.
I'm going to take you back to something you said, and you said some of your inspirations, the big rock stars of microscopy, that you even compared them to bands. When you actually get to know them, they're not quite what they look to be. So, who are your prime examples? No, I'm joking. Don't answer that before you say
No, I'm not.
this person,
I
that
mean,
person.
the idea. No, the idea is that it's I mean, the thing is that once you get more responsibilities, once you get to deal with people, you realize that there's a lot more than just the science, and there are and there are people who say, okay, the science is great, but these are not good people, and people that you say, okay, these people have so many trainees that have become good scientists, So I think this is and I think the system with the flashy papers and everything makes us believe, that is the only thing that counts. And at some point, I think it's important to recognize, the real legacy are the people that will be here after you.
God, you mentioned the drive to publish and the way to publish as being one of the big that makes people look like rock stars to start with, even though maybe they're just very good at playing that system. And actually, on quite a lot of the podcasts that we've done, there's been a lot of people talking about the system of publishing and the difficulties in it. And I've got every sympathy with the publishers as well. But do you think that's going to change? I wish if I could go back to someone doing these sort of podcasts 20 years ago, were they saying the same, or were they, this was the system? But has it now changed and people are trying to game the publications more and pushing more to get in I don't know. Is there a different emphasis? Is there so much volume now that you have to be louder about where you publish? I don't know.
I think so first of all, I think the system has changed profoundly already. between the since I started in, so my first stay in a biology lab was 2001 or 2002, and the system is very different now. And I would say a lot of it is thanks to the preprints and thanks to bioRxiv. And this was not like a revolution that happened in a day. So I think we are not really aware of it. But if you try to imagine and remember how it was before we had bioRxiv, it was a very different system. And now I think it's so, there are still people that don't like it. There are still people that don't use it. But I think most people in healthy communities are really relying on bioRxiv so that science goes faster and in a more open way. And this by indirectly has also changed how the journals are operating, because if you look at how they were really deep against preprints when it was out, and how they are more let's say even pushing for them
Yeah.
now. So this is because their business model has changed a bit because of that. So there was a big change, and I think it will continue. And I think the people that are a bit, there is this discourse of how it was so much better in the 1970s, and how this or that Nobel Prize would never have had a career if the system was like it is now and things like that. I don't really, I mean, first of all, that the system in the 1970s was really an old boys club. So there was absolutely not inclusive, so it was a lot less people. So it was of course it was easier because there was a lot less people, and I would think that the people like the luminaries of that time, or the people that got Nobel Prizes, would have been able to handle the system of today like they were able to. Because if you really want to do science and have something to bring, you will find a way to do it. And in particular, if you are such a genius that what you have will really transform whole areas of science, we'll not say, oh, it's too difficult to publish, I would do something else. I don't believe that.
I would agree wholeheartedly, and it's going to carry on evolving. I think it'll be interesting in another 10 years' time to see
Yeah.
where we're at. If
No,
it
I
is a
think
trend, whether things plateau now, or I don't know.
yeah, I agree. I think the danger now is that we have somehow managed to reduce the importance of journal names. So it is still important, of course, but not as important as it was. But the question is, what do we replace it with? Because if we are, just okay, so no impact factors anymore, no nature cell science anymore. But now, if everyone just look at CVs and say, "Oh, that person was in that big lab, so it's something good," it's not
No,
better. To replace the journal mafia with the lab mafia would not be any better. So we have to be careful that we are we need proxies, we need shortcuts to assess people, but we have to be careful, or at least aware of these bias and these proxies.
so Christophe, on a more light-hearted area, some quick fire questions. Are you an early bird or a night owl?
Oh, night owl, definitely.
Yeah. PC or Mac?
Mac, but not for image processing.
Which is such a change from what it was 20 years ago. McDonald's or Burger King?
Ah, uh, my own burgers.
Okay. Uh, Beer or wine?
Beer.
Oh, I'm from a Frenchman. Wow.
Yeah, I know, I
Ah,
know. I'm
I'm in
ashamed. They
trouble
would, they
now.
would, they would strip me of my citizenship, maybe. I don't know.
Chocolate or cheese?
Cheese.
You passed that one. I've already asked what your favorite food is. Uh, Book or TV?
Book.
Book. What are you reading at the moment? What genre?
At the moment, I'm reading a book on. So I used to read a lot of fiction, and now I'm reading less fiction, and now I'm reading an essay. It's about psychotherapy and how to change. and not only personally, but it's the Palo Alto school, the people that invented a whole area of psychotherapy, and it's an interesting book about what people called I don't remember the exact term, but basically it's about trying to manage situations not by like the first impression of what should be done to change a situation is not always the best one, and sometimes paradoxical actions can have better outcomes than the ones that you would think would change in the right direction. I think it's a very interesting book.
Oh, that's heavy.
Yeah, no, I mean it's no, because it's really it's full of examples, and people they had that have problems in their life, and how by saying going in a direction that was unexpected, they could help solve their situation. And I'm always very interested in these kind of things about how to I don't know how to be better with relationships and with managing people and things like that, because I think that's not my best my best qualities. So I'm always interested in getting better. So
And I think actually the parallels from that, even just in the workplace, even in politics,
yeah.
not just people, but how, yeah, I guess paradoxical, going one way can actually have unforeseen consequences, positive or negative. Uh, what's your favorite film? Favorite movie?
Oh, my favorite movie. So difficult to have one, but I
Give
would
me
say
two.
no. I would say the one that I can watch. It's Alain René, My Uncle from America. You know
Is
that?
what, sorry?
It's Alain René, so a French Nouvelle Vague, and it's called My Uncle from America.
Okay.
And it's not too far from my professional interest, because and also what I just was saying about changes and people personalities, because it's a movie that was made from Henri Labourie's work. So Henri Laborie was a big neuroscientist that worked a lot, and he found the first neuroleptics, I think, in the 1960s. And it's a book about the reaction of rats, when they are in stressful situation, and it's a book about how the way the brain works.
Mhm.
And RNA made a movie that basically exemplifies that with people in situations, and it's kind of exactly bringing what I was talking about, like people and relationship, and also neuroscience, and how you can try to understand that. It's a very good movie.
Do you have a favorite Christmas film?
Favorite Christmas film? Oh, I think the original name is Trading Places, right?
Okay. Yeah, yeah.
I think it's a
Oh,
Christmas movie, right? It says
you're,
Christmas.
you're the first one who's ever said Trading Places.
Really?
Yeah.
No, it's at Christmas.
That, is that Eddie Murphy? Was that?
Yeah.
Yeah.
Eddie Murphy. And no, it's very funny.
Oh, do you know what? I don't think my boys have seen that.
Yeah, no, it's
There you
it's
go, on my
funny.
list.
It's anti-capitalism. It's very good.
Uh, favorite music? What sort of music do you like listening to?
I listen to a lot of I would say jazz, soul, and hip hop. And
Okay.
so I play the saxophone, and I like music a lot. I listen to.
You never mentioned saxophone when we talked about hobbies and what you do outside of work.
So I used to play. I don't play much these days. And actually, I gave the saxophone to my daughter, who started training and playing. So now I get to listen to my saxophone without even playing it. So that's perfect.
Oh, that's almost sweet justice because as she's learning from the very early stages, you'll understand the pain that your parents went through when you were learning at that
Yeah,
stage.
yeah, exactly. I can help it.
Favorite color?
Favorite color? I don't know. Green, I would say.
Oh, you said you listened to some of these. Surely you know it's Alexa 488 or Psi 3 or Psi 5 for your Storm. Oh, Christoph, you missed it. Uh, so I know you from social media as well. I'm following some of your accounts. So Twitter, X, or Blue Sky?
Or blue sky. I'm not on Twitter anymore.
Blue Sky or LinkedIn?
Blue sky.
Ah, it's good to know.
Yeah,
It is.
I mean, I'm still learning, the loss of what Twitter was, and because it has been very important for me and I think for my career, like it was at a time where I was a bit isolated, and it was like it really opened me to a lot of what was happening and discussing science and everything else. And I think no place has replaced what Twitter was. But yeah, I mean, blue sky is not too far. It's still insular. It's still but it's not too far from it.
I found LinkedIn is also vastly changing how it's being used
That's true.
as a
Yeah,
bit as well.
LinkedIn has been. I have started using it more for work and just to but it's more. I think it's more a broadcast system. Like it's not a conversation on LinkedIn.
Yeah. Favorite conference?
My favorite conference, that would be the Neuronal Cytoskeleton Conference in Chile.
Mm-hmm.
So this is a series of conference initially started by Christian Gonzalez in Chile that brought really all the best people in the world that were working on the neuronal cytoskeleton in various places. So this got me to discover Chile. This got me to meet a lot of the people that I consider my community and becoming friends with some of them. And I was lucky to co-organize it 3 years ago. And I think this is really like, yeah, I would say my meeting is that meeting.
We are nearly coming up to the hour. So I'm going to ask, do you have any regrets in your career?
I don't know. I guess I will. My career is not long enough yet.
Is there anything you'd like to go back and change and think, if I was to go back, I wouldn't have done it that way. I'd do it this way. Anything?
Uh, I,
Tough
I guess as always, it has to do with people. I would say there are people that I think, and when you start, for example, and you start managing people, I would say, okay, maybe I didn't manage some people in the way that was the best for them and the best for me, and I was learning, so I would say this is with what I know now, I would be able to be a much better mentor. So because I mean things experiments, things like that. It's just stuff. But people, it's you have only one chance with one particular people. So
Yeah, I think, but you're always learning.
yeah,
That's
that's the most important thing is to never think you are done learning in all areas.
Christophe, we are up to the hour. Thank you very much for joining me today. You've just been amazing to talk to. So thank you
Thank
very
you
much.
very much. I really enjoyed it.
Everyone who's watched or listened, please subscribe to the channel that you're listening to. Please pass it on to your friends, tell your friends, recommend it, because I think there's been some amazing career advice today and some of the difficulties that you can face, and we could get lucky to get the next step or not, and to keep going at it. So, and Christophe has really highlighted some of those excellent elements. And you've also heard him name drop other previous guests, such as Eric Betzig, so forth, that are on there. But Christophe, thank you so much for today.
Thank you very much.
Been delightful, and I'm looking forward to trying your curry at some point if I can invite myself.
Sure. Thank you very much.
Thank you.
Thank you for listening to The Microscopists, a bite-sized bio podcast sponsored by Zeiss Microscopy. To view all audio and video recordings from this series, please visit bitesizebio.com/the-microscopists.